discuss about the complement system. Is it visible, the slide?
Yes, sir.
Fine. Once again, someone is trying to join. So, my dear, I believe all of you have gone through it, the complement system, and I'm expecting that to some extent you should be able to remember back whatever you taught in your classes, regular classes, because many of our students, they were asking to revise the immunology component. So let's start, because it is one of the very important chapter. I thought that let me revisit it. So if we can conclude it and complete it within a short period of time, then I can revise your another chapter on hypersensitivity also. Let's try. So as you know that what is actually complement. Complement means it's a group of protein, a team, a protein group. Just like the many proteins available in the serum, complement as a system, it is nothing but a group of protein. There might be around 30 proteins, and this is serum protein, means all the proteins are available in the serum. So complement system, it works as a group of protein available in our blood. Okay, so in the blood we know there are many proteins available, like when we differentiate them, we can categorize them like the alpha, beta, gamma. So by that way, so this is also a group of protein, a complement protein. Initially, it was first discovered by Jules Bordet. In the year 1893, and during that time he named it as alexin. But now the new term, what we understand, the complement, that is actually given by Paul Ehrlich. So how the complement, this system, the complement concept, people understood, how they have understood what is complement and what is the role, what is happening. Look at that. There are three sets of reaction. It is presented here. Immune serum. What is the meaning of immune serum? Immune serum means a serum.
Excuse me, sir.
Yes, please.
The slide is not moving.
Yeah, that is the problem. So I am leaving that full screen mode. Now is it visible? Can you see this slide?
Yes, sir.
Like
Yes, sir.
concept
Yes, sir.
of complement. So I will go using this mode, all right? Whenever we are making the full screen, then it does not move. Then let us try to use this side. So here, what is happening? Let me try to make it a little bit larger. This will be helpful, maybe. Okay, I can go maximum like this. Here, look at that. There are multiple sets of reactions. In the multiple sets of reaction, what is happening? There is one set. Someone is once again knocking. So what is happening here? Immune serum. What is the meaning of immune serum? Immune serum means, oh no. Immune serum indicates serum containing specific antibody. Take one bacteria. You can imagine about any bacterium. Let's take example, suppose Bacillus anthracis, okay? And Bacillus anthracis, when it gets inside the host may develop the immunity, means in the form of antibody. If we get those serum from that animal, that is immune serum, and that immune serum contains antibody against anthrax, all right? So that means if you have that serum with antibody and mix with that same bacteria, what you can see that at the end, there is lysis of the bacteria. So this is one condition. Another set of reaction, look at this. Non-immune serum means I took the serum from an animal which does not have any antibody. Then we mixed with that same bacteria. At that time, no lysis. Set 3, heat inactivated serum. Heat inactivated serum means immune serum which is heat inactivated. Here, heat inactivation means the temperature is 56 degrees centigrade. When we add that antibody but it is heat inactivated and bacteria, then there is no lysis. Look at this side, first set, same serum, when we mixed with the bacteria, there was lysis. Now same serum, when it is heat inactivated and mixed with the bacteria, no lysis. What is the meaning of that? When we heated that serum, the serum lost its property to lyse the bacteria. Is it okay? But when this heat inactivated serum mixed with normal non-immune serum, then we mix them with that same bacteria, they are making lysis. So here, try to understand the sequence of the reaction. This serum, it is complete serum with antibody mixed with bacteria and made the lysis easily. But when the non-immune, no antibody mixed with bacteria, it could not make the lysis. But when the immune serum is heat inactivated, mixed with bacteria, could not make the lysis. But the magic is that immune serum, which is heat inactivated, mixed with normal non-immune serum, then mixed with bacteria, now I can see lysis. It means two factors are working here. One, we need antibody because it is the non-immune serum. When mixed with bacteria, no lysis. Then in the blood or serum, there are some factors which gets inactivated when we heat it. And that factor, if we can get it from the normal serum, and because it contains antibody, but not that factor which is available here. When we mix them together, all together they are making the lysis. With this experimentation, Jules Bordet came to a conclusion that definitely in the blood or serum, there is some factors which are responsible for the lysis. Basically, that is the set of protein which is complement. So what is that? Complement, what is the role, major role of the complement? So major role, what we have seen, first of all, it helps in production of opsonins. So I am not going back what is opsonins. I believe you can remember back. Opsonin means it is a product which helps in phagocytosis. All right, opsonin are some product or materials or object or something which is available in the system which improves the capacity of phagocytosis by the phagocytes. Then production of anaphylotoxin. Anaphylotoxin, something which is responsible for development of anaphylactic reaction. Then it helps in direct killing of organism. It can, without help of anything else, it can kill the organisms. It helps in enhancing the antigen-specific immune response. Means what we are getting the immune response when antigen-antibody reacts, it helps by this protein. Then it also helps in the maintenance of the homeostasis in the body. So the role of the complement, it is not a single function, what we understand, but there are multiple functions of the complement where it gets activated. Oh, what is that? Complement proteins. What is that complement proteins? As I have already mentioned, that there are around 30 proteins available in serum, and they make approximately 10% of the serum protein. There are many proteins in the serum, so around 10% of them are basically complement proteins. Sources of complement, from where the complement proteins originates in our body. So there are mainly two places where complement proteins generally synthesize in our body. One is the liver, another is macrophage. Other than that also there are many. From liver we get the complements like C3, C6, C8 and complement protein B. And macrophages, so far it is recognized that C2, 3, 4, 5, then B, D, P and factor I. These are the major sources. In other than these names, I mean that terminologies, there are few other names like C1Q, C1R, C1S. You know it, all the name, because already we have covered the complement setup in your regular classes. So I don't think that you have any issue in this name. It is quite understandable. So around 30 proteins, as I have mentioned. But remember, my dear, within the list of 30, probably you have not seen all the name when you read about the activation of the complement at that time. But what are those? Those names are not in the complement pathway. Those are certain regulatory protein. Someone regulates the protein, means the activation of the complement. Someone, if it is having any deficiency, they helps in improvement. Some protein, they are controlling the extra reaction. So all together there are 30 complement proteins. Complement protein, it acts through a cascade reaction. Okay, cascade reaction. Cascade means, we understand that we heard about the name of the cascade, means when we talk about the waterfalls. Cascade fall means stepwise, 1, 2, 3, 4, 5 cascades, 2 cascades, 3 cascades, means stepwise jumping. Complement system, it is a series of protein, and their action, it gets starts with one trigger, then it can activate one protein. That same activated protein can activate another protein, then the same second activated protein can activate the third protein, then third one can do the fourth protein. That means it is a series of reaction. The complement protein 3, complement protein 4, or complement protein 5 cannot be directly activated single-handedly. What does it need? It needs the help of its previous protein. Okay, so that is the cascade. Means when the cascade starts, you need to start with some of the reaction, then it will go on, just like the one pathway of the blood clotting in our system. The blood clotting gets starts with the any type of injury, releasing of the protein, then one by one all the factors get start to activate, and at the end we get the fibrin formation. All right, so activation pathway, I talked about, the involves a cascade of successive completion. Then it enhances a small initiating signal. Small initiating signal means who will get start this reaction. Then the components are cleaved into activated fragments. Complement proteins, they are available in our system, all are in inactivated stage. Okay, all are in inactivated stage. They are dormant, they are available in the serum, they are not in use, but whenever there is a need, then they get start. So the question mark is, how the complement protein understand that when our turn is here, when we need to get activated. So that is the signal, or that is the initiation, which is known as a trigger reaction. Means just like releasing one bullet from one rifle, there should be something like a trigger. If we push the trigger, bullet gets off. Means because complement system is like that. It is a very dangerous bullet. So whenever there is a trigger, reaction starts. So the fragments, it induce intense inflammatory response to eliminate infectious agent. Complement system is very deadly type of system. When they are dormant, there is no issue. When they get activated, the activated complement will always destroy the target. So we need to handle it with care. So that handling system, it is the regulatory system. Let's get started. Then what about the complement activation? Okay, now I want to go back little bit about the basics of immunology. I think I am audible to all, everyone. Fine. And if anyone having any doubt in between, please raise questions. I can restart. So before talking about this complement activation pathway, I want to go back to the basics of the immunology. So far, all of you understood that what happens when a foreign body enters in our body. Foreign body, all right? Foreign body enters in our host system. I am talking about human body, it may be animal body also. We understood so far that our body develops immunity, correct? Immunity in the form of either humoral immunity or cell-mediated immunity. There is no doubt so far about it. When we have developed, suppose, humoral immunity, we say antibody developed. Development of antibody, after it develops, they are available in the serum or in other body fluid. Then what happens? These antibody can bind with the target antigen. Okay? So the target antigen, it may be a bacteria, it may be virus, it may be a cancer cell, it may be anything else, whatever the foreign body. Fine, now the antigen antibody complex formed. But question is that, think all everyone, please try to think about when the body responded against the foreign body, antibody developed, that antibody now binds with the target antigen, it formed an immune complex. Up to that is quite fine, everything sounds good. Major question starts now, what is the fate of this immune complex? Whether this body will carry, whether our body will store it in our system. So it should be removed from the body, obviously, because immune complex is equally dangerous just like the normal foreign body available in our system. So whenever there is formation of antigen antibody complex, next step, removal of these objects from the body. A bacteria, when it binds with an antibody, the bacteria will not die. A virus, it binds with its antibody, the virus will not be killed. It should be killed, it should be removed. So who do this job? It is the complement system. When there is formation of antigen antibody complex, next step, you have to kill or because the antibody does not have the capacity to kill the target. Antibody can only hold it, bind it. Next job, it is done by complement system. This is the story. So that's why complement is such an important component of the immune system. This means, so now we need to understand how the complement system gets activated. So there is 3 different pathways by which complement gets activated. Now, complement activation, it is having 2-step reaction. First step, activation of complement protein 3. Activation of complement protein 3, C3. Until or unless complement protein 3 gets activated, it cannot go to the next step. So basically, the first step, it is the initial phase or enzymatic phase. Here, there are 3 options by which complement protein 3, the C3, can be activated. This is classical pathway, alternative pathway, and lectin pathway. All the 3 pathways, what they can do, they can activate the complement protein 3. After that, there is a common pathway, that is the terminal pathway. Terminal pathway means it ends the reaction. Ends of reaction is what? Objective is simple. It will destroy the target. The target may be a bacteria, a virus, a parasite, a fungi, anything. But initially, our body has the capacity to activate the complement protein C3 by 3 different mechanisms. One is known as classical pathway, another is known as alternative pathway, another is known as lectin pathway. Out of these 3 pathways, classical pathway, it is antibody dependent. Antibody dependent means for the activation of this pathway, we need the help of antibody. Means this is the acquired immune response. Alternative pathway and lectin pathway, these 2 pathways does not require any help of antibody. They can be activated independently. So they are the part of innate immunity because it does not need any help of our antibody or anything else. So these are the parts of innate immunity. This classical pathway is a part of the acquired immunity. Okay, now once by these 3 different pathways, we got the activated C3. Next is the terminal pathway get start. Now we will discuss today about the activation of classical pathway, is formation of antigen antibody complex. So this is the activator of this pathway. So this is also known as a trigger reaction. Means who initiates or who puts the trigger for classical pathway, it is the antigen antibody complex. So whenever this activation start, this activated the formation of soluble antigen antibody complex or binding with antibody, then to a target cell. Then the actual reaction starts. Let's see what actually happens in the classical pathway. I have two graphical presentations here. This is one chronological reaction. This is one image-based reaction, okay? Image-based presentation. In classical pathway, there is involvement of 4 complement proteins. 4 proteins are C1, C2, C3, C4. Let me repeat. In classical pathway, 4 proteins, C1, C2, C3, and C4. Okay. But first protein is our C1. C1 protein, this is the picture of C1. C1, it is like a bundle of sticks. At their top, you can see there are knob or ball type structure is available. What this C1, now C1 protein, it has 3 components, C1q, C1r, C1s. Let me repeat, the C1 protein or C1 complement, it is actually composed of 3 components. C1q, I have one better picture here. This is the C1, complete C1 protein. This is the C1q, the main part. It is having 6 sticks and all their head, it is just like a knob or a dumbbell type. So this is having 6 sticks, a bundle. This is C1q and 2 more dumbbell shaped structure. This is the C1r and C1s. They can be placed within the hands of this C1q. So altogether, this is a C1 protein. Now look at this head of the C1q. This head of C1q actually placed in the antibody molecule. In the antibody molecule, there are certain receptors available. This is known as complement receptor. All of you, I think you can remember back antibody molecule and their structure. All right. And it is having a receptor. This is known as complement receptor, means where complement can bind. Look at this. This is the surface of the target antigen. This is the antibody molecule binds. Here, the head of the C1q binds with those antibody. Means when the antibody binds with the antigen, then this antibody gives the attachment with C1q. This C1q then call the other 2 proteins C1r, C1s. When this complex completed, this is the activated form of C1. In the normal serum, there are free C1q, free C1r, free C1s every time available. This C1q used to search where is the antigen antibody complex. Whenever there is antigen antibody complex, then this C1q can easily bind with the complement receptor of the antibody. When they bind with the complement receptor of antibody, then C1q and R and C1s, they can bind with them. When this complex formed C1qRs, then it becomes activated. The C1 now activated. When C1 activates, they can activate next complement protein. The next complement protein is C4, not 2. It is not in sequence, my dear, remember. C4, when this C4, suppose if it is a ball-like protein, then what happens? When it comes in contact with the activated C1, then it breaks down into 2 portions. One major portion, this is C4b, and one small portion is C4a. Whenever this C4a and C4b remains together, this is inactivated C4. Let me repeat. C4b and C4a, when they're joined together and they remain inactivated. If by someone this C4b and C4a can breaks down and make it in two portions, then this is activated form. C4b is the activated complement of C4, and this C4b now binds with C1q. Means C1q it can bind. So now C4b is the activated protein. Okay, C4b is the activated protein. Next, if C4b becomes activated, next job is it binds with next complement protein C2. Everyone try to understand. If you have doubt, please raise your voice. We can repeat. So C4b, if it is activated one, immediately binds with C2. Immediately binds with C2. Then, once again, with the help of activated C1, this C2 also similar protein, just like the C4 inactivated protein, then it breaks down into two components, C2a, C2b. The C2a is the activated component, then it makes a complete C4b, C2a complex. C4b, C2a complex, and small fraction C2b, it release into the serum. Next job is the third protein, it is C3. C3, I told that this is the target to activate the complement protein C3. Now it is a classical pathway or alternative pathway or lectin pathway, doesn't matter. Our target is to activate the complement protein C3. So it forms the complex C4b, C2a. This protein complex, we can activate the next protein, complement protein C3. Where C3 is inactivated, similar fashion, when it comes in contact with this complex C4b, C2a, then it can be activated by dissociation into C3b and C3a. C3b is the active molecule. C3a is released. Then it forms C3b. So our first objective to activate the C3 is fulfilled here. We have started with the antigen antibody complex. When there was antigen antibody complex, it could allow the C1 to be activated. How it gets activated? The C1q molecule binds with the complement receptor of antibody. This C1q can bind with C1r, C1s. So this is the activated C1 molecule. When C1 gets activated, it can act on another protein. This is C4 molecule. This C4 molecule is inactivated one, but when it comes in contact with C1, activated C1, it becomes activated. How? By dissociation into two components, C4b and C4a. C4b is the active molecule, C4a released. C4b now binds with C1. Then this activated C4b can bind with C2. Okay, can bind with C2. After binding, now C1 can act on C2 again. So C2 breaks down into C2a, C2b. C2a is the active molecule. C2b is released in serum. So now it forms a complex C4b, C2a. This complex now can react on complement protein C3. C3 once again activated by breaking down into two components. One component is C3b, which is the activated molecule, and C3a is released. So finally, we have seen the complement protein C3 is now activated. Now look at this. There are small components which are being released into the serum. This is C4a, C2b, C3a. So these are not used in the complement activation system, but they are being released in the serum. But this is fact that in the physiological system, nothing is wastage. All these molecules which are released at the time of activation of complement like C4A, C2B, C3A, these are sometimes acts as anaphylotoxin, these are sometimes acts as opsonins. These are sometimes act as the C-reactive proteins. They are different times. So they are always being used for some of the other reactions. Okay. Now, this is the activation pathway of activation of complement by classical pathway. Anyone wants to ask anything up to this?
Sir,
Yes, please.
sir,
Yeah. Benya.
sir, C4, C4B will bind with C42 with help of C1.
C4B, C4B,
Binds with C2,
C2,
C2.
C2. Yes. Because this is the complex, you see, the activation is based on the first step is activation of the C1. Okay. C1 then acts on C4B. Is it okay? Then when the C4B will bind with C2, full C2, it is not activated. That means it is C4B, C2. Then this C2 is activated with the help of C1 again. Look at this arrow. This C1 helps to activate the C2 again. This C2 will be broken down into C2A and C2B. This C2A is the active molecule, C2B is released. Then it forms complex C4B, C2A. Is it your question or something else you are asking?
No, sir, this is the question. Thank you, sir.
Oh, is it okay? All right. Anyone else? If no, then I can proceed. All right. Fine. I think all are satisfied so far. Then let me continue with this. Here, same thing, another diagram, okay? Just to make it simplified, I have drawn here antigen antibody complex. AB complex means antigen antibody complex. Here one, there is the activation of the C1Q, C1R, C1S. Then it is the activated antibody antigen complex and C1, which acts on the C4. Then C4B formed, C4A as anaphylotoxin released. C4B now it binds with C4B, acts on the C1, C2. C2 is broken down, C2A and C2B released as the kinin-like activity protein. C2A, it form a complex. Look at that C1, 4B, C and 2A. So this is the C3 convertase, which acts on C3. We are getting C3B. So the complex C4B2A, C4B2A, this is a complex. It is known as classical C3 convertase. Classical C3 convertase because it can convert C3 into activated C3B under the classical pathway. I have already described this picture about the C1. Now, one C1 complex, C1 QRS complex, this is the electron micrograph picture. I think it will be appreciated. It is the original electron micrograph picture that indicates the 6 head of the C1Q and complex of the C1QRS. Here, all of you please try to understand one important part. We have given the structure of the antibody and we have given the structure of the C1 molecule. Classical pathway, so far I think it is now understandable that classical pathway gets start with the formation of antigen antibody complex first, then binding of C1Q. Is it okay? Now look at that, the structure of C1Q, it has 6 heads like this, and it is a fixed molecule. To activate the C1 molecule, minimum 2 heads, minimum out of the 6, 2 heads should bind with the complement receptor of antibody. Let me repeat that C1 molecule is available free in the serum. It does not get activated normally. How it gets activated? If minimum two of the heads of C1q binds with the complement receptor of antibody. So that means suppose this is one antibody, this is one antibody, if they place together side by side and two heads of the C1q can bind with them, then only it will be activated. Now question is that suppose this one is placed here, little bit away. Now one head is bound, but another head could not. This will not be activated. But now if it is placed too close, that only one could attach, another could not, still it will not be activated. So question is that, so two molecules of the antibody, they should be placed in such a way that they are getting minimum two heads bound with it. Only then our C1q can be activated. Is it okay, or everyone? So it is conditional. Binding of C1q with the antibody, it is very crucial. It is not random. It is not as per their will. It can bound only when they are located at a perfect position. If it is far away, it cannot. Although anti-antibody complex formed, but still C1 is not activated. Too close, they cannot. Means on the surface of this antigen, when they place at the perfect position, they can bind. They can bind and they can continue the activation pathway. But in case of the disturbance in the positioning, it cannot bind. So because we understand that IgG molecule, IgE molecule, IgD, all those molecules, they have having a monomer, like monomer means single molecule. So in case of IgG, there should be minimum two IgG molecules required, and that too should be placed in a very perfect position to activate the C1q molecule. Is it okay? In case of the IgM molecule, no, IgM is a pentamer, correct? Pentamer means they are always 5 molecule, every time it binds. So whenever they are placing on the surface of antigen, it is a single molecule with 5 components. And our C1q can easily placed nicely on the IgM molecule without any problem. So in the IgM molecule, one IgM is enough to activate our C1. But for IgG, how many molecules required? Minimum 2. But that too conditional. They should be placed at the right position, not away, not too close. So now question is that in case of the classical pathway, which class of antibody is better, IgM or IgG? For activation of classical complement pathway, which class of antibody is better, IgM or IgG? Do you like anyone to answer it? IgM, always. Yeah, exactly. Now the reason I have tried to explain why IgM is always better than IgG to activate the classical complement pathway. The actual fact is lying here, the attachment of C1q molecule. C1q is very sensitive. It needs the perfect positioning of the antibody to get access the complement receptor on it. IgG is a monomer, so there should be minimum 2 antibodies placed at a perfect distance so that they can give the access to the C1q. Whereas IgM is already pre-built, fixed, and their positioning is very nice, where C1q can easily place itself and can get activated. So a single molecule of IgM is always more than enough to activate the C1, but it is not true for the IgG. Now I am not talking about the IgD or IgA or IgE. What is happening here? IgD cannot activate the classical complement pathway. Can anyone say why? IgD. IgD is not eligible to activate classical complement pathway. Why? Can you remember back the structure of the all antibody molecules? IgG, IgM, IgE, IgA, IgD. Out of these 5, IgD cannot activate the classical complement pathway. Can anyone answer it? Why?
Sir,
Yes,
sir, because
please.
it is monomer and it is very low in amount.
No, my dear, it is not the actual fact. It depends upon the structural activity. IgD molecule, if you can remember back, IgD molecule does not have the CH2 domain. CH2 domain. Right now, I don't have the picture with me. CH2, I mean let me go show the picture. Antibody. I think it will help. Look at this structure of the antibody. This is the heavy chain, okay? And the pink and orange is the light chain. And in the heavy chain, this is the what domain? Light blue. It is, can anyone please first, light blue color, this is which domain? Variable heavy, VH. Then CH1, CH2, CH3. Okay? CH1, CH2, CH3. The complement receptor is available in the CH2 domain for all the proteins, not for IgM and IgE because they have extra domain. In case of IgD, this CH2 domain is missing. IgD molecule, the CH2 domain is missing. Means they don't have a complement receptor. If there is no complement receptor, then where the C1q bind? So IgD, as it does not have the CH2 domain, so there is no complement receptor, so it cannot activate the classical complement pathway. Another thing is that IgE class and IgD class, these are the membrane or the cell bound antibody. Because IgE class and IgD class, they are not available free in the serum. They are available always attached on the surface of the cells. Which cell? Basophil and any other cell? Can anyone say? On the surface of which cell IgE and IgD are available? One is basophil. Another cell, can anyone remember back? Related cell, basophil. Another is a cell which activates the type 1 hypersensitivity. Which one? Which releases lots of vasoactive amine, causes inflammation. Which cell? Related cell, basophil and mast cell. Mast cell, all right? Basophil, they are the membrane bound. So IgD cannot activate classical pathway. Reason I have explained, because it does not have the CH2 domain. As there is no CH2 domain, there is no complement receptor. And out of this rest 4, best one is IgM, then IgG, then IgE, then IgA. IgA is another sometimes it is a monomer, sometimes it is a dimer. Where you will get it as a dimer, when it will get it as a monomer. Come on, I think most of you will know it. You can just recapitulate your memory. IgA is available as a monomer as well as a dimer. In which situation, in which position, in which location you will get it as a monomer, and where you will get it as a dimer. Am I audible to all? Am I audible?
Yes, sir. Yes, sir.
Yes, good. Fine. So you cannot remember back, right?
Sir, in
IgA,
zero.
When serum means circulation, they are monomer, means single molecule. When they are available in the secretion, they are as a dimer. This dimer is known as secretory IgA. Secretory IgA. Why it is secretory IgA? Because they release in the secretion and they have a protection with them. Suppose IgA is available in the intestinal tract. Is it correct? IgA is available in the intestinal tract. In our intestinal tract, they are available. They are protecting our gut. IgA is available in our nasal secretion. IgA is available in the sometime in our urine also. But remember, these are the location which is very dangerous for the protein like antibody. Antibody is nothing like a protein. When these proteins are available in our intestinal tract, suppose what will happen? There are lots of digestive enzymes, acids, alkalis are available. So whenever IgA will reach to the intestine, the digestive enzyme will immediately digest the antibody also. So there is no logic. It is simple fun to be there, but it does not happen. What happens? When the IgA reaches to the intestinal tract, before reaching, they make a dimer. Two IgA molecule binds together with a small molecule. This is known as secretory molecule. This secretory molecule is a protective shield for the antibody which protects them against the proteolytic digestion of the any kind of enzyme. So this is the form of the secretory IgA. When it forms this double bind or dimer of the IgA, once again they cannot bind with the complement receptor. Okay, so in that time, secretory IgA also will not do, but the monomer can do it nicely. I believe you can remember now. You should not forget these few points. All right. So let me proceed. I think we don't have too much of time. I need to finish this part. This part just now I have explained it, and I have shown the IgM molecule and its superiority in the activation of the classical pathway. Now, anyone, if you wants to ask anything out of this,